Good morning and welcome to the University of Regina. My name is Andrew Cameron and I'm going to talk to you a little bit about some of the research that happens in my lab in the Department of Biology and the Faculty of Science here at the University of Regina. I'm going to share my screen with you to provide some illustrations of just some of the work that we do in biology. In the Department of Biology, we cover, well, the breadth of biology. So we study the natural world and we're very interested in the environment, ecology, evolution. We study human, animal, plant health, ecosystem functions and how all these pieces fit together and much, much more. And within our department, new students and all students get to enjoy hands-on labs. There's field work to be done. There's undergraduate directed research projects where undergraduate students can go into research labs and spend a semester or more doing that hands-on research. There's a Bachelor of Science Honors Program that provides even more exposure to research in the process where you get to lead your own research project. And then after completing undergraduate studies, there are graduate studies as well. And our department is a very rich environment full of many, many graduate students doing a very wide array of different types of research projects. So a little bit about the research in my lab. One of the things that I work on as a microbial geneticist is the interest of how microbes, all the invisible things like bacteria and viruses around us, how they connect different features of our environment. So here, an urban human, myself, when I go for a walk with my dog in Grasslands National Park, what microbes might we interact with in that wild prairie ecosystem? Which microbes do we leave behind while we're there? And which microbes can potentially cause us disease is one of the big questions that we address. And collectively, this whole understanding, this network of connections between microorganisms and animals and plants and humans, we tend to call One Health. And we're interested then in how do changes in different environments, so perhaps climate change and how it's going to shift the amount of moisture and temperatures in that grassland ecosystem, how can that potentiate the evolution of new pathogens, both for plants, animals, and humans, and how can they pass between these different animals and plants that can occupy that system? As a microbial geneticist, me and my lab, we use DNA sequencing as a major piece of our toolkit to understand both which microbes are in our environments and on our bodies, and then how they evolve and how they spread, how they can become pathogens, how they can become antimicrobial resistant. So much of this information lies in their DNA. And what's really neat about how fast these technologies of DNA sequencing are developing is that what you see here are small little benchtop sequencers, and we have both of those in my research laboratory that allow us to, they're portable. We can sequence DNA outside of the lab now. We can sequence an immense amount of DNA within the lab to reconstruct what these microorganisms essentially look like on a genetic level and be able to track them and understand their interactions with their ecosystems and how they evolve. Now, in studying pathogens and these relationships between animals and environments, I do this both in Saskatchewan and around the world. So for example, I'm involved in multiple research programs in Southern Africa, and you see there a picture of the Chobe River in Botswana, where me and my group, we study the relationship between wild animals in that park. So everything from elephants to lions, that water system and that water that feeds downstream communities and how those communities can either become infected or produce new pathogens that then can spread into wildlife. We also look at similar systems here in Saskatchewan. So we're very interested in how antibiotic resistance evolution and different pathogens that can impact cattle can spread in agricultural systems and how water availability can impact that transmission. We also work across Western Canada to understand things like tick-borne diseases, and those occur both in the forests of BC that you see illustrated there and on the prairies of Saskatchewan. Increasingly, because of climate change, ticks are able to spread into new parts of Saskatchewan and with them bring diseases. And one of the most effective ways to detect those diseases and understand them is through microbial genetics and DNA sequencing. So I run a research lab, and I'm also the co-director for the Institute of Microbial Systems and Society. And here you see a pre-COVID picture of most of the members of our group gathered. And our group is composed of undergraduate researchers, graduate students, postdoctoral fellows, and research associates. And we are funded by many different agencies in Canada and from abroad that help us study health, both for humans and animals and plants. And mostly in my lab, we focus on human health and interactions with pathogens. And that's what I'm going to tell you a little bit about in the next couple of minutes. So for example, one thing we're very interested in is antimicrobial resistance. And this is a growing problem that continues to spread around the world. And in this graphic, you can see the estimate of how many people will die directly from an untreatable microbial infection by the year 2050. And this is a huge burden on health. The number of people dying from antimicrobial resistant infections is expected to exceed the number of people dying by cancer within the next few decades. So what do we do to address this? Well, I've already said that I'm a microbial geneticist, so we can use genetics to do many, many different things. On the top left-hand corner there, you can see how we can reconstruct and understand where outbreaks and ultimately pandemics start from, and then how they spread from individual to individual or between different animals or plants. And in that upper left-hand corner there, you see evidence collected from studying the genomes of SARS-CoV-2, the virus that is currently causing the COVID pandemic. On the upper right-hand side there, you can see another aspect that we take in my lab to understand infectious disease, that we're very interested in how does ecology influence how organisms become either pathogens or harmless to us. And for example, we study the interaction between E. coli and Salmonella, two microbes that could encounter each other in our intestine and many, many other microbes there. And they can make us very sick, or they may not. They may live in a passive or benign state that does not make us sick. So we're interested in what about our system, the things that we eat and the other aspects of our environment might incline those organisms to become pathogenic in our system. In the bottom left-hand side there, you can see that by sequencing genomes and genes within these microorganisms, we can track down the genes that cause features we're interested in, like antimicrobial resistance. And when we find those genes, then we can start to, again, in the lab, dissect what is the mechanism by which that organism is able to resist that antibiotic. And ultimately, that can greatly inform treatment options. So for example, with that set of genes that you see in the bottom left-hand corner, when we discovered those by sequencing genomes in our lab, we were able to tell clinicians exactly which genes were involved. And the diagnostics that used to take 6 months to detect what type of antimicrobial resistant infection was in that very sick person infected with this bacterium, now that test can be done in a day because we know exactly which genes to look for. And in the bottom right-hand side, we also study in my lab how genes move between microorganisms. So we study both the mechanisms and the consequences of genes jumping from one species to another and how that can influence general ecology, but also specific features like antimicrobial resistance and pathogenesis. And to wrap up, I'm going to tell you a little bit about some of our work currently on COVID. So in my lab, we do multiple different research projects on the coronavirus. And as geneticists, of course, we're very interested in how does this virus both evolve and how can we develop better ways to detect it and track its evolution so that we can both identify and anticipate variants of concern. So there you see an illustration of SARS-CoV-2, the virus that is causing us so many problems. We know that originally it came from a bat, but it may have jumped directly into a human from a bat. It may have gone via another mammal and then into a human. It may even have gone via a lab and then jumped into a human. Those are all possible origins of the current pandemic. Now, when that virus did jump into a human back in late 2019, as illustrated by that red dot, since then it has spread to millions and millions of people. And as it spreads over time, it accumulates mutations. And we can track those mutations. And those mutations are important because they create new variants like alpha and delta that are so impactful for our health. And here, what you're seeing is over time, how those mutations are accumulating and changing the SARS-CoV-2 genome. So each one of those dots represents a different virus isolated on a different continent. And we can see that over time, there are more and more mutations. And within there, there's a few dots that represent the Delta variant and its derivatives as well. Now, the original virus that jumped into a human probably doesn't exist anymore. It's mutated so much now that we don't have the original SARS-CoV-2. What we have is many, many thousands of different variants circulating around the world and continuing to generate new variants. One big project that we're doing is to work directly with public health labs to try to develop better techniques to both sequence and identify those variants, but also detect what co-infecting viruses and bacteria might also be in the human host. And so here that human is actually infected by potentially 2 viruses. So what happens when a person contracts both COVID and the flu? It's probably very, very bad, but we don't know what is the risk of that happening. So we need better ways to detect these different viruses. What if a person has COVID and an antibiotic-resistant streptococcus? How is that maybe going to impact disease and inform treatment decisions? And then the other types of things that we can do is ask questions about how many different coronaviruses circulate in nature. So for example, we work with collaborators in Central Africa who send us samples from bats, and we're able to sequence the coronaviruses in those bat samples and understand coronavirus diversity and how they evolve, and ultimately not only understand how the coronavirus is currently evolving as it spreads in humans, but what other coronaviruses have the potential to jump to other animals and to humans. So this is really a tricky thing to do in the lab, because when we take a sample like that nasopharyngeal swab that is taken if it's testing someone for a SARS-CoV-2 infection, so they've gone in and had that swab stuck up their nose, to find the pathogen is tricky because most of the genetic material in that swab is from the human host and from other healthy or benign bacteria that live in the respiratory tract. And that's represented by the needle. And the virus that we want to detect is literally that needle in a haystack, because it's a tiny, tiny, tiny amount of genetic material. So how do we detect it and sequence it? Well, there are already technologies being used daily around the world to track SARS-CoV-2 variants. What we're working on is developing perhaps the next generation of that detection method, where we can extract that genetic material, and instead of amplifying just the SARS-CoV-2 to detect it and sequence its genome, instead we're using a way to capture all the different viruses that might be in a human respiratory tract, and we're going to add bacteria so that we can capture just those genomes, the genetic material that we want to, and then put it onto our benchtop sequencers that we have in my lab, that we also are used regularly by public health labs, both in Saskatchewan and across Canada. And we have found that this technology works very well. We can sequence the whole genome of SARS-CoV-2. We can find those variants, understand the transmission. We can track how those variants are spreading across Saskatchewan, for example, by generating this whole genome sequencing. We can find novel coronaviruses, and we've been able to find, for example, not only novel coronaviruses, but novel spike proteins that exist in different coronaviruses to better understand how that important component of the virus changes over time and really dictates the interaction between the virus and its host. And we can also detect with these technologies other viruses and other bacteria that are infecting the human host. So that gives you an overview of the types of research that we do here at the University of Regina in the Department of Biology. And this research, this project, these multiple projects I've just described on coronavirus, we have undergraduates who are contributing. They're analyzing that genome sequence to identify things like spike proteins. We have graduate students who are also working on that project, and we have postdoctoral fellows and research associates who work on that project in my lab and in our institute. So very nice to meet you. Have a great welcome week, and we'll hopefully see you around campus.